In the name of Allah, who is the most merciful and the most beneficent. Assalamu alaikum. I am Hina Fatima, student of MS Chemistry, Batch 5 at the University of Management and Technology. And in this video, I'm going to talk about enzyme reactors, trickle bed reactor, and the packed bed membrane reactor. And I have done this assignment under the supervision of Dr. Hamad Majid. So in this video, I'm going to talk about these reactors in detail. Firstly, we're going to discuss about the enzyme reactors, the basic general introduction of the enzyme reactor, and then their scientific background, definition, and then I'm gonna answer some important questions. And then the types of the enzyme reactors, which basically includes batch reactor and then I'm going to talk about construction and working of the batch reactors, then their merits, demerits, and then the second type of the enzyme reactor, which includes continuous stirred tank reactor, their merits, demerits, working, and their application in chemical industry. A reactor is a very basic term that is used in chemistry. So a chemical reaction in which two reactants stick together and they form a particular product. So the vessel, container, or the device in which the particular chemical reaction is carried out is called a reactor. And the chemical reactants, they are designed in order to increase the efficiency of that reaction which is carried out in the vessel. So in this video, I'm going to talk about the enzyme reactors in detail. Firstly, we're going to discuss about their scientific background. So as we all know, enzymes are proteinaceous in nature and they are biological catalysts. So there was a person called Edward Buchner who discovered that the enzymes are also capable of catalyzing a chemical reaction even with outside the living cell. So that was the basic foundation of the enzyme reactor. So talking about the introduction of the enzyme reactor, an enzyme reactor is a specialized device or a system that is designed for the controlled and the efficient use of enzyme in various biochemical and chemical processes. So there was a definition, that was the definition of the enzyme reactor. And enzyme reactors are vessels that are employed to carry out the desired conversion of raw materials with the help of enzyme, and that enzyme can be immobilized or not during the process. Now the important question is what is called immobilization, or why it is important to make an enzyme immobilized. Immobilization is simply just restricting the movement of the enzyme molecule. So in enzyme mobilization, it's a basically process in which the enzyme molecule is confined to a solid support over which the substrate are passed and the reaction carried out, and they are converted into the product. In enzyme reactor, the stability of an enzyme is a very important factor. So in order to carry out the reaction in an organized manner, it is very important to make an enzyme immobilized. So the basic aim of the enzyme reactor is to allow the enzyme and the substrate to come into contact to each other, and in the enzyme reactor, both the enzyme and the substrate both are placed in the separate compartments, and then they bring together in the proximity of each other to proceed the reaction. So the purpose of the enzyme reactor is basically to maximize the reaction efficiency via compartmentalization or bringing the reaction counterparts in close proximity to each other. So there are the types of the enzyme reactors. Number one is the batch, and the second is continuous stirred type of reactor. So in the batch reactor is a very simple kind of a reactor in which there is a closed vessel in which a particular chemical reaction is carried out, and it is a type of a non-continuous reactor in which the reactants feed into the reactor at once, and then we're going to talk about their design and working. So in batch reactors, a batch reactor is generally consist of a tank which contains a stirrer, and there are some big fixed baffles that improves the stirring efficiency. Or in other way, we can simply say that the tank consists of an agitator, and the purpose of that agitator is to mix the reactant together so that the reaction carried out with smooth efficiency. So if the reaction is exothermic, the batch reactor consists of some coils. So in case the reaction is exothermic, these coils will cause the cooling effect, and if the reaction is endothermic, then these coils will cause the heating effect. And the batch reactor is a non-steady transient reactor. It means here the extent of conversion within the reactor, it depends on time. Then there are some key points, like as you can see in the graph, there's a red line which shows the concentration of the substrate, and the dotted line which shows the concentration of the product. It means that both the substrate and the product, they are removed In a batch reactor, after a fixed period of time, all the product gets removed, and which means both the substrate and the enzyme molecule, they have the same residence time within the reactor. Then there are some merits. Talking about their merits, so the some number one is their simplicity. That's why they are used for small scale production of highly priced products. And the second is their controllable environment. As we are working in the form of batches, so it is very important or easy for us to control on the entire process. And the greatest advantage of operating a batch reactor is its versatility, as the batch reactors are especially used in cases where the reaction produces lots of product. And they are also used when continuous operation of a process proves to be difficult due to the viscous or the interactable nature of the reaction mixture. With a few advantages, they also have some disadvantages or demerits. And the number one or the major demerit of the batch reactor is their operating cost. The operating cost is very high because we have to work in the form of batches, so we are supposed to empty the entire reactor, and then we refill the reactor for the next batch, so it will increases the operating cost. And the second is disadvantage or demerit is their non-productivity, and then there could be also batch to batch variations within the batch reactor. And the another disadvantage is there a lot of labor, as we have to empty the reactor and then we have to refill it again, so it requires a lot of labor depreciation. So, talking about their use, batch reactors are basically used in labs to study the kinetics of the liquid phase reaction system. Now we have other type of enzyme reactors, which are continuous state tank reactor. It is also known as CSTR reactor or MFR. MFR means mixed flow reactor. In this reactor, the reaction occur in a closed vessel or a tank, and it consists of a agitator. And the purpose of that agitator is to mix the reactant thoroughly, so that the reaction can go with higher efficiency. And it is different from a batch reactor in the sense that the name itself indicates it is a continuous type of equipment. It means in this reactor, the process is carried out in a continuous manner, not in a batch manner. So how does a CSTR work? A continuous tank reactor is a container used for chemical reactions. It allows the substance needed for the reaction to flow in while the product flow out at the very same time. So in this reactor, both the reactant and the product, they move at the same time within the reactor. This makes it a great tool for making chemical continuously. The CSTR reactor mixes the substance well and works consistently under steady conditions. Typically, the mixture that comes out is the same as what's inside. So the flow rate of the product or the reactant will be the same in continuous state type of reactor. In certain cases, when a reaction is too slow or two different liquids are present, they requiring a high agitation rate, a CSTR can be connected together to create a cascade. Now, is a CSTR a batch reactor or not? It means no, a CSTR is not a batch reactor, as the name is also showing the same thing. So the main difference between the CSTR and a batch reactor is that a CSTR is a continuous flow reactor, while the reactants are continuously feed into the reactor and product are continuously removed. But in the batch reactor, a fixed amount of reactants are added to the reactor, and then they allow to react until the reaction is complete before the products are removed. So in a CSTR, the reactants are continuously mixed by using a stirrer or agitator, which ensures that the mixture is homogeneous or well mixed. And that is why the CSTR are used in large-scale industrial processes where the continuous supply of reactant is required. On the other hand, batch reactors are used in laboratory scale experiment where smaller quantities of reactant are required for testing and analysis. Now there are some key points. CSTR is a steady state equipment. It means the extent of conversion here in CSTR, it does not depend upon time. And the reactant and the product, they both enter the vessel at the same mass flow rate. And then we have some advantages. So, biggest advantage of using a CSTR in industry is that it can produce a large amount of products, and being a continuous steady state reactor, the reactor can keep on operating on its own end. And the second is temperature control is easily maintained, and then the CST behavior is well understood, including in mixing, ability to handle, the reaction calorimetry, and the other is it is less expensive and it is easiest to construct than dedicated specialty flow system. And it can also use for PAT, process analytical technology. There are some disadvantages. The major disadvantage is that the CSTR cannot be used for reaction which have very slow kinetic, because it will require a reactor of a very large volume. A steady state needs to be maintained, so the system needs to be well understood. And then the fabrication and the operational cost of the reactor may make it infeasible, and batch reactor is used in this case. Single units are not optimal for reactions with slow kinetics. These are the some disadvantages or demerits of the continuous state type reactor. Then there are some applications of the enzyme reactor, and the enzyme reactors are widely used in various fields due to their versatility and specificity. Number one is biotechnology and pharmaceuticals. So the enzyme reactors are used in the production of pharmaceutical, biofuels, and specialty chemicals. They enable the synthesis of complex molecules with high selectivity. The second application is their food and beverage industry. Enzyme play a crucial role in food processing, such as the production of cheese, bread, and beverage. Enzyme reaction improve the efficiency and quality of these processes. Then the third application is their environmental remediation. Enzymes are utilized in environmental application to break down pollutants and contaminants, because these enhances the efficiency of bioremediation processes. They are also used in biofuel production. Enzyme reactors are used in the conversion of biomass into biofuels like ethanol and biodiesels. Then it is also used in the wastewater treatment, and here it can improve the efficiency of biological wastewater treatment process by breaking the organic matter and reducing the pollutants. Enzyme reactors are also used in clinical diagnostics. Enzyme reactors. Now our second topic is trickle bed reactor, and the basic history of the trickle bed reactor, and then the introduction of the 2-3 lines, the basic definition and the explanation why they are named trickle, then the trickle flow regime, and then we will discuss the construction and the working of the trickle bed reactor, then merits, demerits, and their application in chemistry. Talking about his history, in the early 20th century, the groundwork for the trickle bed reactor was laid with the studies on fluid dynamics, and then in the 1920s, Lewis Ferry Richardson was the person who was working on catalysis, and then they led the stage for later development. And then in the 1950s, many scientists has observed the behavior of the reactants flowing through the packed catalyst particle, and this laid the foundation for the trickle bed reactor concept. Talking about its introduction, so it is considered to be the simplest reactor type for performing catalytic reactions, where a gas and the liquid are the two reactants, and the reactants are fed into the reactor, and further the reaction proceeds. The trickle bed reactor, the liquid moves downward to the catalyst surface, and the gas moves in a counter current motion. So trickle bed reactors are solid liquid gas contacting devices wherein a liquid stream flows downward over a pad of catalyst with a pressure difference which will serve as a driving force. The fluid or the liquid flows over the catalyst particles, and they form the thin film or droplets. The gas streams can either flow co-current with the liquid or in a counter-current motion to the bed. Trickle bed reactors are primarily operated in continuous mode, but they are sometimes can be used in a semi-batch process. Why they are named trickle? So the trickle bed reactors are named as such for their operation in a trickle flow regime. This regime is characterized by stable and continuous flow of the liquid and gas streams to the bed. Similarly, the laminar flow is a single phase system. Trickle bed reactor can also be run in pulsing spray or bubble flow regimes depending on the application. So basically we can say that in trickle bed reactors, depending on the liquid and the gas flow rates, there are the four flow regimes that have been followed. Most important and the most followed flow regime is the trickle flow regime. So trickle flow regime occurs when both the liquid and the gas have low flow rates. In this flow, the liquid flows down the reactor on the surface of the packing in the form of rivulets and film, while the gas phase it will travel in the remaining void spaces. So the trickle flow regime can further be divided into the two regions at a very low gas and liquid flow rate. When the flow rate of both the gas and the liquid is low, the liquid flow is laminar. In laminar flow, both the liquid and the gas phase molecule, they will move in a parallel direction to each other. And the second region is when the fraction of the packing remains unwetted, and this kind of regime is called partial wetting regime. And if the liquid flow rate is increased, then the partial wetting regime will changes into the complete wetting trickling regime, and in which the packing is totally covered by a liquid film. And this flow regime is also termed as the complete continuous regime or the homogeneous flow or low interaction regime, because very little interaction between the gas and the liquid exists in this regime. The diagram is also shown where the liquid moves over the film and the gas will move through the void spaces. Then talking about its construction. So the construction of the trickle bed reactor, it consists of a reactor vessel. The main body of the trickle bed reactor is a vessel or a column that is often made of stainless steel or the other materials that is compatible with the reaction condition. And this vessel hold the catalyst bed and provide the necessary structural support. The catalyst bed inside the reactor vessel is typically made of solid material that will facilitate the desired chemical reaction, and is often present in the form of small pellets or granules. Then there are some inlet and the outlet ports are also present inside the reactor. Inlet is used for the introduction of the reactant, gas or the liquid into the catalyst bed, and the outlet is used for the collection of the product. And the purpose of these ports is ensure the proper distribution of the reactant across the catalyst bed. The distribution system is also present inside the reactor, and the purpose of that distribution system is to ensure the uniform flow of the reactant to the catalyst bed. And this may include the distributor plates or the nozzles to evenly distribute the liquid and the gas over the catalyst bed. Because of this distribution system, the reaction occurs in an organized manner. And then there is a working of the trickle bed reactor, which involves the introduction of the reactants. The reactant, basically the gas or the liquid, are introduced into the reactor, where the gas and the liquid may contain the substance that need to undergo a catalytic reaction. Then the trickle flow, unlike the system where the liquid flowed over a catalyst bed, in a trickle bed reactor, the liquid flows down through the catalyst bed in a continuous trickle, and this ensures a more controlled interaction with the catalyst. Then there occur a catalyst. Liquid moves down to the catalyst bed. It will come into contact with the catalyst particles, and then the catalytic reaction will occur at the surface of the catalyst that will lead to the conversion of the reactant into the product. These products are then removed from the reactor with the help of the outlet port, and depending on the nature of the reaction and the product, separation processes may be applied to isolate the desired product. Trickle bed reactor, they also allows a continuous separation. In this, it also allows for a steady flow of the reactant and the removal of the product. And this continuous mode is beneficial for different industrial processes, and there are several advantages of the trickle bed reactor. The absence of the moving parts in the trickle bed reactor result in a lower maintenance cost. So the maintenance cost will be low, as because there are no moving parts. And then the plug flow operation and the effective catalyst wetting result in high reaction convergence, and then the liquid holdup is less because the ratio of the liquid to solid is less, which reduces the possibility of homogeneous side reactions. And the trickle bed reactors, they also have a larger capacities, and they can be operated at a high temperature and high pressure. And the problem of flooding is not encountered in because of the concurrent downward flow operation. And because of the trickle flow regime, trickle bed reactor also provide the efficient mass transfer between the liquid and the gas phase. The concurrent flow of the liquid and the gas over the catalyst bed allow for intimate contact, promoting high reaction rate and improved efficiency. This trickle flow regime also minimizes the temperature gradient within the reactor, and this feature is beneficial for managing the exothermic reactions and maintaining the optimal temperature conditions. While the trickle bed reactors have several advantages, they also come with certain drawbacks, and here are some of the demerits or disadvantages associated with trickle bed reactors. And the number one is the channeling and the bypassing. In trickle bed reactor, there is a risk of channeling because when liquid tend to flow through the preferred pathway in the catalyst bed, this can lead to uneven distribution of the reactant, and it will reduce the contact with the catalyst, and that will at the end impact the overall efficiency. And the second is the pressure drop. Trickle bed reactor may also experience relatively high pressure drops due to the resistance encountered by the liquid as it passes through the catalyst bed. When the liquid will pass through the catalyst bed, it will face some resistance, so it can result in a high energy consumption for pumping and can limit the reactor's scalability. The continuous trickling of the liquid over the catalyst bed can also cause mechanical wear and attribution to the catalyst particles, and this may result in the generation of fines which can affect the reactor performance and require additional separation steps. Trickle bed reactor continuous removal of the heat is difficult, so this can cause damage to the catalyst in highly exothermic reactions. And then the catalyst effectiveness is also reduced due to the large catalyst size particles. Trickle bed reactors are less effective with viscous and foaming systems. They have limited applicability. These reactors may not be suitable for all type of catalytic reactions, and the choice of the reactor depend upon the factors such as reaction kinetics and the nature of the reactants. And there are some applications of the trickle bed reactors in the chemical industry. First one is in hydrogenation process. So these reactors are utilized in the hydrogenation of vegetable oils to produce edible oils, and this process involves the addition of hydrogen to the unsaturated fatty acids. The second is petrochemical industry. They are also used in the petrochemical industry, playing their role in hydrotreating processes within the refining of petroleum. Hydrotreating process will remove the impurities from the crude oil fractions, and it will improve the quality of the refined product. And the third is the chemical synthesis. These reactors are also employed in various chemical synthesis reactions, such as the production of the fine chemicals or specialty chemicals, where precise control of the reaction condition is crucial. They are also used in water treatment process for pollutant removal, and the catalytic reaction within the reactor can help in breaking down and transforming the contaminants. They are also used in biochemical processes, particularly in biorefinery application, where they can contribute to the conversion of biomass into biofuels or bio-based chemicals through catalytic reactions. Trickle bed reactors are also used in environmental remediation process, where they facilitate the catalytic reaction to break down the pollutant and contaminant. So the versatility of the trickle bed reactor stems from their ability to handle a wide range of catalytic reactions that make them valuable in process spanning different industries. Now moving on toward our third and the last topic of our today's video, that is packed bed membrane reactor. So the packed bed membrane reactor, it combines the principle of the packed bed reactor and the membrane technology in order to enhance the chemical processes. And in this type of a reactor, a solid catalyst is packed in the reaction zone, and a membrane is employed that will cause the separation of the reactant and the product from the catalyst. And this innovative design offers several advantages for certain types of reaction. Here are some key features or the benefits of the packed bed membrane reactor. Number one is enhanced selectivity. Packed bed membrane reactor provide enhanced selectivity by selectively allowing certain molecules to pass through the membrane, as this is particularly useful in reaction with simple product where separation is challenging. The membrane in these reactors will cause the separation on the basis of the particle size. Further, the presence of the membrane will also facilitate the efficient mass transfer that will ensure a higher contact surface area between the reactant and the catalyst, and it will lead to the higher reaction rate and the higher conversion efficiencies. Ability to selectively transport the specific molecules, so the membrane can also minimize the side reaction that will lead to the improved reaction specificity and purity of the desired product. And the fourth one is the controlled reaction condition, in which the membranes can help control reaction condition by selectively allowing the passage of certain reactant or product. This control can be crucial for optimizing reaction kinetics and improving the overall reactor performance. The presence of the membrane also improves the heat management. They allow for better temperature control by separating the reaction zone from the rest of the reactor and preventing the excess heat buildup. The major advantage of the packed bed membrane reactor is the reusability of the catalyst. The packed bed design allow for the use of the solid catalyst, which can be reused. The membrane helps to retain the catalyst within the reactor while allowing the reactant and the product to pass through. These kinds of reactor are versatile, and they can be employed to various types of reaction, including hydrogenation, dehydrogenation, and other catalytic processes. Despite these advantages, it is important to note that designing and operating the packed bed membrane reactor can be complex, and their feasibility also depends upon the specific characterization of the reaction involved. So the packed bed membrane reactor is a complex yet efficient system that combines packed bed reactor technology with membrane separation in order to enhance the chemical processes. And here is an overview of the construction and the working principles of the packed bed membrane reactor. So talking about its construction, it consists of a catalyst bed, and the reactor contains a catalyst bed that is typically solid, made of small particles or the pellets. And this bed is designed to provide a large surface area for the catalytic reactions. It also consists of a membrane module, and the membrane module is integrated into the reactor. And this module includes a selectively permeable membrane, and that membrane will allow the certain molecule to pass through while inhibiting the passage of the others. There is a reaction zone. The catalyst bed and the membrane module are situated in the reaction zone, and this is where the catalytic reaction takes place. And the membrane assists in separating the reactant and the product. Then there are some inlet and the outlet ports. Inlets are provided for introduction of the reactant into the reactant, and outlets are designed to remove the product or for the collection of the product from the reactor. These ports are strategically positioned to ensure the proper flow through the catalyst bed. The temperature and the pressure control. The reactor is equipped with mechanism to control pressure and temperature that will maintain the optimal reaction condition for achieving the desired outcomes. And there are some working principles of the packed bed membrane reactor, which involve involve the introduction of the reactants into the reactor. These reactants could be gas or liquid that contain the species involved in the reactions are introduced into the reactor. Then there is a catalytic reaction system. As the catalyst flow through the catalyst bed, the catalytic reaction occurs on the surface of the catalyst bed, and these reactions could involve chemical transformations such as hydrogenation or dehydrogenation. Simultaneously, the selectively permeable membrane allows the certain molecules to pass through while blocking the others, and this separation occurs based on the molecular size, charge, or other specific properties of the component. The molecule with a smaller size will easily flow through the permeable membrane, while the molecule with a larger size will get entrapped in the pores of the membrane. Similarly, the molecules with a greater charge will flow fastly through the bed or the reactor, while the molecule with a low or a small charge will get entrapped in the reactor for a long period of time. The product of the reaction which have passed through the membrane are collected at the outlet, and the separation of the product from the reaction zone it help minimizes the undesired side reaction and improve the product purity. Depending upon the specific application, unreacted component or the byproduct will again recirculated to the system for the further processing in order to enhance the overall efficiency of the reaction. The integration of the membrane also allows for the better control of the reaction condition, which includes the temperature and the concentration gradients that will contribute to the reaction efficiency, reaction selectivity, and the output. Then there are some demerits of the packed bed membrane reactor. Here are some potential drawbacks associated with the packed bed membrane reactor. So the basic demerit of these reactor is the membrane fouling. One significant disadvantage is the potential for membrane fouling. Over time, the membrane may accumulate contaminants, reducing its efficiency and requiring the periodic cleaning and the replacement. Fouling can also be caused by the deposition of the reaction product, catalyst particle, or other substances that are present in the feed. A packed bed membrane reactor can be complex to design and operate. Integrating the membrane system with the packed bed membrane reactor requires careful engineering to ensure proper functionality. And further, the monitoring and the controlling both catalytic reaction and membrane separation, it will add complexity to the overall system. The major disadvantage is the cost. The cost of the membrane material and the complex design can make these reactors more expensive to set up, and then their maintenance will be difficult. So the membrane technology in particularly can be costly, impacting the economic feasibility of certain applications. Reactors are not suitable for all types of reactions, so their effectiveness depends upon the specific characterization of the reaction. And in some cases, the benefits gained from membrane separation may not be overweight the associated cost and complexities. Membrane may experience wear and tear over time, especially in harsh chemical environments. So the durability of the membrane material is crucial for maintaining long-term efficiency and avoiding the need for frequent replacement. And also the major drawback of this reactor is the pressure drop. The presence of the membrane can contribute to the increased pressure drop within the reactor system. Managing pressure differential is important for maintaining optimal flow rates and ensuring the efficient reactor performance. Despite these advantages, it is important to note that the feasibility and the success of these reactors depend upon the specific requirement of the reaction and the desired outcome. So, advances in membrane material, reactor design, and operating strategies continue to address some of these challenges, making these reactors a promising technology for certain application in the field of chemical engineering. So, the packed bed membrane reactor find application in various industrial processes where a combination of catalytic reaction and selective membrane separation can offer several advantages. Number one is hydrogenation and dehydrogenation reaction. So these reactors are commonly employed in hydrogenation and dehydrogenation processes. The selective membrane allow the removal of hydrogen that will prevent the backward reactions, and it also enhance the conversion rates. These reactors are used in industries such as petrochemical and chemical synthesis. Also, these reactors are used in fine chemical and pharmaceutical industry for the utilization of reaction that require precise control over reaction condition and separation of product. This include processes like chiral synthesis, where enantiomer selectivity is important. Then they are also used in biodiesel production process, particularly in the transesterification of triglycerides to produce the biodiesel, and glycerol membrane separation helps in removing methanol and other reactant that will improve the overall reaction efficiency. Kinds of reactors are also used in esterification reaction and then environmental application. With the separation of water or other byproduct via membrane technology helps derive the equilibrium towards product formation. And in environmental application, for the removal of pollutant from wastewater and the treatment of the industrial effluents. The catalytic oxidation processes, oxygen is introduced to oxidize the certain compounds, and these reactor enhances selectivity of the oxidation reaction, and membrane separation helps in selectively removing reactant or product. They are also used in natural gas upgrading by removing the impurities such as sulfur compound, and the selective membrane enable the separation of sulfur containing compound, contributing to the production of cleaner natural gas. Then here are some references. Thanks for watching.